right, so last time we were talking about glucose catabolism, right? The different pathways that you could break down glucose, and breaking that down into pyruvate and then getting energy and some reducing power. Then we talked about essentially two things that could happen once you break down the glucose. You can either go into fermentation if you don't have a terminal electron acceptor around, or you can go into the TCA cycle. So here's some review questions from last time. I heard somebody say an answer. I'm generally hearing that you are saying C, but yes. So so the pentose phosphate is the only one that you get only NADPH and not NADH. Do you remember which one gives you both? Yep. All right, one more review question. Anybody give me an answer for this one? Yep. So just remember the trick here is that you will need to see what it's asking per, right? So the numbers that I gave you last time were either per pyruvate or per glucose, but the question here is asking you per acetyl-CoA. So just be aware that depending on what you're talking about, they can enter at different stages in the cycles, okay? All right, so today we're gonna essentially complete the story of what happens for catabolism for, at least we're talking about just your standard going from NADH to O2, and we'll go through the electron transport system and the proton motive force. So this is just an overview of where we have been so far and what we've been talking about. So it's been, well, a couple of weeks ago now, because we talked about this before spring break. We had started with catabolism, right? Talking about food sources and how those get broken down, different hydrolysis reactions, and you end up with glucose, and in a lot of those reactions you end up with acetate. The acetate can go, and we talked about this last time, can get directly sent towards the TCA cycle. And then we also last time talked about the 3 different ways you can break down glucose, right? But essentially what you end up with is pyruvate and then some variation of energy and electron carriers. So now what we also talked about last time was, so TEA means terminal electron acceptor, and so in the absence of a terminal electron acceptor you get fermentation, and then through the TCA, with the terminal electron acceptor you would go through the TCA cycle. The idea for both of these is that we want to start regenerating the NAD plus and the NADP plus, right? And so now essentially we're here at the end of the TCA cycle. We ended up with, again, more ATP and then more electron carriers. So now we have to figure out what's going to happen with these. And so that's where we're going to go through today, is this essentially. So the electron transport system, your books call it the electron transport system. Some of you have probably heard of it as electron transport chain, depending on what book you're using. And then that leads to a proton motive force, and that can then be used for several different cellular functions. All right, so the electron transport system, again, so we talked about this a little bit before, right? That life transfers energy by moving electrons. And so electrons move, so talking about the whole process back to that kind of workflow chart that I was just showing, electrons moved from reduced food molecules to energy carriers, right? And then, so now in the electron transport system, we have membrane protein carriers, and we'll talk about what these are today in class. And then so the electrons kind of move through these, and then finally to oxygen or oxidized minerals, so which in this case is what the terminal electron acceptor is. So the electron transport system is a series of membrane protein carriers that yield energy through successive redox steps. So remember, redox is oxidation reduction reactions, right? And through these different redox steps, you're actually generating a proton motive force that stores energy that allows the cells to make ATP. So this is basically the story that we're going to be telling today. So there are actually several types of metabolism that use ETSs, and so you can have the organotrophy or chemoorganotrophy. So organic molecules don't need electrons, so again, this is like glucose, right? And then it can be either aerobic or anaerobic, meaning the terminal electron acceptor can be oxygen, or it can be something like nitrate. Other examples, so the lithotrophy, so these again are inorganic substrates, remember, versus the organic substrates. Again, you can also, in these cases, still have aerobic versus anaerobic. And then phototrophy, the light absorption actually excites the electrons, so you're not necessarily getting them from glucose. The light itself will excite the electrons. This is usually coupled to splitting of either water or hydrogen sulfide, and we'll talk about that in a couple of classes. So regardless, if electrons are donated to an ETS and ultimately reduced to a terminal electron acceptor, this is what we refer to as respiration. All right, so now just a kind of a reminder of your redox reactions. So redox reactions, the delta G values are proportional to the reduction potential, so this E value. So this is between the oxidized form of a molecule, which is the electron acceptor, and its reduced form, or the electron donor. So the reduction potential represents the tendency of a compound to accept electrons. Okay, so that's important to remember. And it's usually measured in volts or millivolts. So now in this case, you want to remember that positive values of E have negative delta G values. And so essentially that means that gaining electrons yields energy. And so this is the equation. So again, remembering that this not and prime refers to standard conditions and 1 molar concentrations. In this case, N is the number of electrons and F is Faraday's constant. All right, so here we have what we call the electron tower of standard reduction potentials. So we have the electron acceptors in this kind of pinkish red on the left column, and then the electron donors in blue. So a redox couple, so this is the oxidized and reduced state of a compound. So one of these is a redox couple. The more negative values represent stronger electron donors. So we talked about NADH, right? That's the one that is holding that electron and moving it around the cell, right? So that's going to be a stronger electron donor. So these have the more negative values. And then the more positive values represent stronger electron acceptors. So we are always talk about how oxygen is the terminal electron acceptor, right? So oxygen is down here is the positive values. Directly from here. So we have that equation here is written here, and the values written there are these values here. Back, and we look, so here's that equation, right? But we want to flip it around because we want the electron donor from the NADH side. And so you just remember these values coming from here. These can be, these ones are always written in the oxidized and reduced states are always in one direction, but you can flip them, right? Because most of these the reactions are reversible, right? So you can give electrons and it can also take electrons, and so it's just flipping. Okay, so now, but a complete redox reaction, you have to combine two of the individual redox couples. And so one that accepts electrons from the red column and one that donates electrons from the blue column. And so the reduction potential for the overall reaction is given by adding the reversed reaction of the donor to the electron acceptor. So we'll walk through this for the example of NADH, oxidation of NADH by oxygen. And then, so first we want to do the electron acceptor. So the electron acceptor in this case is going to be oxygen, right? So I'm just going to show you that this equation came. And so that's what we have here. And then again, so now what we want to do is for the overall reaction, we want to add the reversed reaction. So the reversed reaction of the electron donor, right? So in this case, the electron donor is NADH. And so if we go, so we flip that equation around, and then so we also put a negative sign on the reduction potential value. And then all you do is simply add them together. And so this here, so again, you see this equation, if we're used to talking about the delta G value, so this is a negative delta G, right? It gives you energy. And so here is the overall, the overall reduction potential for this complete redox reaction. Any questions about this? Yeah?
What does it mean when it says reverse reaction again?
So reverse, just flipping it. Any other questions? Okay. So now we'll get into a little bit about the actual ETS. So this functions within a membrane. So the proteins are membrane embedded proteins. And so it's going to be in a membrane that separates two aqueous compartments. So remember that gram negatives, we have two different membranes, right? So, but in this case, in gram negatives, the ETS is always in the inner membrane. So it separates the cytoplasm from the periplasm. Some respiratory bacteria actually pack their ETS within other structures called lamellae. And so again, but it's always a membrane that you can embed these proteins in. So the idea is that an ETS transfers electrons onto a membrane-embedded carriers in a series of increasing reduction potential ending at the terminal electron acceptors. So essentially, it's going to get these electrons and it's going to pass them along in increasing reduction potentials and eventually ending at the terminal electron acceptor. And we'll see a few more, break this down a little bit more as we go through. And then the other key is that some of these steps actually yield enough energy to pump ions across the membrane. And so that again is how we will get the proton motor force and eventually how we actually get the ATP out of this whole process. Okay, so now getting into some of the proteins themselves. So the proteins are often referred to as oxidoreductases. So these oxidize one substrate while reducing another. So essentially what these proteins are doing is they're those reactions that we were just looking at on that electron tower, they're essentially allowing those reactions to come together and happen. So they're bringing those two half reactions together and bringing the components together and allowing those redox reactions to happen. They will actually often compose of multiple proteins, so that it's more than it's a big complex of proteins, and so there can be more than one protein involved in the oxidoreductase. One of the components is often called cytochromes. So these are colored proteins that absorb visible light. The absorbance spectrum shifts when there is a change in the redox state. So here's the absorbance spectrum for one of the example cytochromes. And so again, you see, so the dashed lines here, so this is what it was originally, and then it's shifted in the reduced state here, and so it has this shift. So again, so different species of bacteria can make many different cytochromes with different absorption peaks, and so they can absorb at different wavelengths, and which also converts to different amounts of energy. They will pass electrons sequentially from each protein complex to the next stronger electron acceptor. So again, so here's just kind of an example. If we were taking electrons from NADH and it passes to this cytochrome, and then this one can pass it on to the next cytochrome, and then eventually there's more steps in the ETS, but this is just an example. Eventually it's going to pass the electrons to oxygen. But again, so this is in terms, it's always passing something into the next higher reduction potential, right? So kind of the idea behind this is that it's always easier to make smaller steps, right? It's easier for me to pass something off to somebody that's very close by, rather than and then you can pass it off to somebody next that's also just really close by, rather than if I had to get it to like over there and I just decided to try and throw something, like the chances of me actually throwing this over there accurately and then that person getting it is much lower than if we just went hand it off little by little, right? So that's kind of the idea behind these passing off of electrons. And then also, if I were to like throw something really far, I'm just going to waste energy that way, right? And so you lose some energy that way. There's less loss of energy if you're making smaller transitions. And so that kind of leads into what the cofactor part of the protein complexes that are playing a role in these transfers. So again, the ETS proteins, the electron transfers they want to do in small reversible energy transitions. So again, like I just said, it's better if it's a small handoff than a large one. And so to enable that, the proteins often will have cofactors. And so these can be small molecules that are associated with the proteins. So some of the examples are heme, flavin mononucleotide, and ubiquinone. And then small energy transitions, they're typically involving metal ions, so such as iron or copper. So this is an example of an iron so these are often will be coordinated with the amino acid residues within the protein. Or they're conjugated double bonds and the heteroaromatic ring. So we had talked about that, right, with NADH, the ring structure, and that's how it actually is able to accept the electrons. Another example here is in the ubiquinone. So again, these conjugated double bonds and in these ring structures that allow it to accept these electrons. But again, all of these things are kind of helping with the small energy transitions. Okay, so respiratory ETS contains at least 3 different components. So the initial substrate oxidoreductase or dehydrogenase, which is here, a mobile electron carrier, and then a terminal oxidase. So we're going to go through an example for this full process. And so we're going to go through the example of going from NADH to oxygen. And the examples will all be, in this class, we always use E. coli as the first example, and then later on we'll learn about some of the other alternatives. Okay, so for the initial substrate oxidoreductase, so the 2 electrons we're going to get, we're using NADH as the example. So the 2 electrons we're going to get from NADH, this essentially came from catabolism, right? So this equation here in the picture, we got this from the catabolism. And so that is going to get passed on to this oxidoreductase, which is called NADH dehydrogenase. In this case, it's NDH1. So there are 14 different subunits in the E. coli version. And so this is the whole complex here, this yellow part here, this is the whole thing. But you can see, even within here, you can see the different subunits that they've kind of drawn out here, right? So there are 14 different subunits within this one protein complex. And so within the complex, right, each of the cofactors hand off electrons to each other through adjacent connections. So it's going to come in here, and one is going to hand off to here, and it's going to keep handing it off. And then eventually, the electrons are transferred to a quinone. And we'll talk about that in a second. So within this example, so within NDH1, oxidation of the NADH, it yields enough energy to pump up to 4 hydrogens across, so 4 protons across the membrane. So certain ones of these energy transitions allow you to pump across a proton.
So the quinone acceptor is located inside of NDH1?
No, it's a little bit misleading. It's going to come up right up next to it, and it'll be a little bit more clear later. So essentially what it's thought of is for how these protons are getting pumped across the channels is that there are amino acids. So the channels are proteins, right? So there's amino acids sticking out, and essentially the proton just kind of hops along adjacent amino acids, and then until it makes its way out of the membrane. All right, so the next part is the mobile electron carrier. So this is the quinone pool. So the quinone can receive 2 electrons from the substrate oxidoreductase along with 2 hydrogens from solution. So the quinone, so if you put the 2 electrons on there, so in order to balance the charge, it needs to pick up 2 hydrogens. If not, it won't be able to like sit in the membrane, right? And so now in this picture, we represent this by Q being reduced to QH2. So the quinones can diffuse within the membrane and carry reduction energy to other ETS components. So in here, it kind of looks like, oh, there's just maybe this one quinol that's kind of passing it off to each other, and maybe that would happen. But it could also be the case that this is in the membrane and it's just kind of moving along until it hits the next protein. Okay. So after transferring the 2 electrons to the next protein complex, the 2 hydrogens are released. So the 2 protons are released, and they're usually released onto the other side of the membrane. So essentially, this part also pumps 2 protons. All right, so this ends at the terminal oxidase. So the terminal oxidase, again, it receives the electrons from the quinol, so the QH2, and it transfers them to the terminal electron acceptor. So this usually includes a cytochrome that accepts the electrons from a QH2. So there's usually a cytochrome here, and the cytochrome is bound to an oxidase complex containing a series of electron transferring carriers. So again, within the terminal oxidase, there's going to be a couple of electron transfer steps, and then in that case, there are at least one transfer that can pump protons through the energy transfers. So the example that we have drawn in here is this E. coli cytochrome BIOL315 oxidase. So in this case, each oxygen atom receives 2 electrons. So at the end, it's going to pass the electrons on to oxygen, right? And it combines with 2 H+ from the cytoplasm to form water, and so that's shown here. But in this case, the 2H+ consumed balances out the 2H+ released by catabolism. So back when we had to break down the molecule, when you made the NADH and plus H+, there were 2 H+ from that, and so these H+ kind of balance those out. All right, so I just talked to you about the kind of common complexes in the ETS, but bacteria can produce several alternative protein complexes in each case. So not only, say, can E. coli produce other forms, but different bacteria, another bacteria might have a completely different one also. And so examples of these, so the initial substrate oxidoreductase, so you can, instead of NDH1, you can have NDH2. So this, again, it does the same job of transferring the electrons to Q, but in this case, it doesn't pump any protons, and that energy is lost during heat. And so E. coli will use this during rapid growth, essentially. And then as far as the mobile electron carriers, so they're different oxidoreductase complexes will interact with different quinones, and so they're just different kinds of quinones, like ubiquinone or menaquinone. And then another example of a terminal oxidase, so they can actually vary in the number of cytoplasmic protons that are pumped. So this cytochrome BD quinol, it still does the same job of reducing oxygen to water, but in this case, it doesn't pump any extra protons across the membrane. But so the advantage of this one is that it can actually bind O2 at much lower oxygen concentrations. So if there's lower O2 available in the environment, then E. coli will likely switch to using this BD quinol. All right, so here's a question for you all to think about. So what is the advantage of the oxidoreductase, so this guy, transferring electrons to a pool of mobile quinones? So why doesn't it just pass the electrons directly to the cytochrome complex. So take a minute, think about it. You can talk to your neighbors or whatever, and then we'll see what you can come up with. All right, anybody want to share some ideas? Why might this be the case? Yes, Roland.
Would it use less energy if it were, rather than going straight for this electron?
What do you mean use less energy?
Or would require less energy to pass on this electron? Or
Yeah, so the gap between the energy transfer might be smaller. Yep. Any other ideas? Yep.
it moves more hydrogens out of the cell.
Yeah, so we get the bonus of getting one extra or pumping more protons out. Yeah. Any other ideas?
Does that cause a gradient?
Sorry, what?
Does that cause a concentration gradient?
For the protons?
Yeah. So like
Yep.
it would be easier.
Yeah, so we're going to get to in a little bit exactly what we do with the gradient, but yep, it does.
Are electrons polar and the membranes are not polar inside, so it needs it to move?
Sorry, what?
The electrons are like polar, and the inside is not polar, so it needs membranes to move.
Yeah, so if there was this big gap here, electrons can't just go. Yeah, it needs to have the contact with something to go. Yep.
Wasn't it what you were saying earlier about the likelihood of reaching its destination? You're having it off instead of just like throwing it?
Yep. Yeah, so it's kind of same idea. The electrons can't really just go through the lipids, right? It has to. So in that sense, right, if the two proteins would actually have to be like right next to each other also in the membrane, right? But yeah, and also that's also related to what Roland was saying about how the energy gap might be huge between these two. Any other ideas? Okay, good. Yeah, those were all very legitimate reasons why this happens. Any questions about this? Any more questions?
What would be the right?
I think those are all correct answers, right? Yeah. So, like, to me, legitimate reasons that would be that again, the space, like, the membrane space, but then also the more, like, if you think about it from the energy efficient reason, is being able to do the small handoffs. If not, you would imagine that the different protein complexes would have to match their energies, and right, remember that these are made up of different proteins and have different cores in each one, so they would have to, the cell would essentially have to put more regulation into making sure that they have the right kinds of proteins in their membrane. All right, so just as a comparison to mitochondrial electron transport, so again, mitochondria also have this electron transport system, but they generally have a single ETS, which is optimized because these are now intracellular, and the environment inside the cell is much more uniform than, say, the environment that bacteria, the membranes of bacteria might experience because of the environment changing on the outside. But they do have homologs of bacterial ETS components, but they do differ from E. coli in the sense that there is an intermediate cytochrome oxidase complex, so there's an extra complex you notice in the mitochondrial version. The mitochondrial version also pumps more protons per NADH. And then, of course, you would imagine that the protein complexes have subunits that are more relevant to eukaryotic proteins than bacterial proteins. All right, so this has been leading us up to the proton motive force. And so the chemiosmotic theory states that the energy from the electron transfer between the membrane proteins is used to pump protons, which is what we've already been talking about. It accumulates in a higher H+ concentration in the compartment outside the membrane. So this essentially generates the proton motive force, this delta P, which stores energy that can be used to make ATP. So if you remember now, so H+ can only flow back through proton-driven complexes, through a membrane complex. They can't actually diffuse through the membrane, right? So it needs something to come through in order for it to get back in. And so it will require something like the ATP synthase for it to come back through. But as a side effect, so this also generates a charge difference across the membrane, right? So protons carry charge with it, so it's also creating a charge difference. So the concentration difference plus the charge difference will makes the proton potential. So that they're all related by this equation here. And when you estimate RT and F for standard temperatures, it's about 60. So the electron potential, so this arises from the separation of charge between the cytoplasm and the solution outside the membrane. But remember, protons aren't the only thing that's around inside and outside of the cell. So the charges from other ions such as potassium and sodium can actually contribute to this electrical potential. And then in the same case for pH, so the pH difference is the difference between the pH internal and external. Again, it's directly changed by the proton potential because that's essentially what we were measuring in pH, but it's also affected by the generation of acids or pH changes outside the cell. So both of these things are directly affected by pumping protons, but remember that there are other things that can lead to charge or pH changes. All right, so now if we kind of remember and think back about what we just learned about the ETS, so there are 3 different fates of the protons in the ETS. So the protons are pumped across the membrane by the oxidoreductase complex, right? And so that directly contributes to the delta P. Protons are consumed from the cytoplasm by the quinone, and then when it's released, right, when the quinone transfers the electrons, it's released to the outside. So again, these protons again contribute to the delta P. And then, but in the last step, right, protons are consumed by combining with the terminal electron acceptor. But in this case, if this balances the protons that were released by catabolism, then it doesn't actually affect the overall delta P. All right, so now that we have a proton motive force, what can the cell do with it? That can drive several different cellular functions. We talked about flagellar rotation, so that's one thing that it can use to the proton motive force for. And again, it can also use it for efflux, so the different pumps that we had talked about a while back, those can also use the proton motive force. But what we're going to talk about now is how the cell actually generates ATP from it, so using the ATP synthase. Okay, so ATP synthase, so you'll also see it written as F1, F0 ATP synthase. So that's this complex here. So the F1 and the F0 complexes rotate relative to each other, so within the membrane it's going to rotate. The F0 translocates protons across the membrane. So this is the F0 portion, the part that's in the membrane. And it actually has this channel here where the protons come through. And then the F1, it consists of 6 alternating alpha and beta subunits surrounding a gamma drive shaft. So this middle part here, you imagine it actually goes down the whole, this whole portion here. That's the gamma portion. And then there are alternating alpha and beta subunits that surround it, so in like a circle kind of formation. So each third, so the alpha plus beta, can convert ADP plus inorganic phosphate to ATP plus water. And so this now is called this whole process is now what we call oxidative phosphorylation. So the H plus pumped by the ETS actually drive the phosphorylation of ADP to ATP by ATP synthase. So here's just more of a cartoon version and kind of breaking down what it actually looks like. So the flux of 3 protons through F0 is coupled to forming 1 ATP. So 3 protons come through here, and this essentially starts the turning. And then as this turns, it causes these conformational changes and essentially is what allows the ADP to come in and the inorganic phosphate to come into the different compartments. And then as they come together, that's how it gets converted to ATP. And so I posted the video on Canvas. It just animates essentially what this mechanism is doing. But just a note to remember that generation of ATP is reversible through this process. And so ATP hydrolysis by F1 can actually pump protons back through the membrane. So if for some reason the cell is like, I really need to take in nutrients and it needs to use the proton motive force to send some stuff into the cell, then it can actually, and it has enough ATP inside the cell, it can use that ATP to regenerate the proton motive force and then use that to take in nutrients. All right, so that brings us through this process here, essentially. So now we've kind of talked through the whole process of catabolism, but only for your standard like E. coli using oxygen as the terminal electron acceptor. So next time what we're going to do is talk about some of the alternate electron acceptors, and we'll also go into the phototrophy next time. That's pretty much it for today.